morning, I am Andrea, member of the Agave Alchemist team, and I'm going to present the results we obtained throughout 10 weeks of working on the experimental design of agave tequilana micropropagation. Agave tequilana is one of the most emblematic plants in Mexico and one of the most important plants in the industry since it is extensively cultivated mainly in Jalisco to produce tequila, being a fundamental mainstay of this state and country's economy. However, although not very representative, other products have been produced from this plant such as sugar, cosmetics, medicine, and biofuels. Regarding the main biological characteristics of this plant, its leaves grow in a rosette pattern, their seeds are classified as monocots, and they die after seed production. They also produce an important amount of metabolites. Primarily, they produce sugars, hydroxycitric acid, which acts as an enzyme inhibitor to avoid the production of acetyl coenzyme A, palmitic acid, a saturated long-chain fatty acid, pyrrole, which has a role in chlorophyll synthesis, and mannitol, which helps in the metabolization of reactive oxygen species. As for secondary metabolites, it produces rutin, which avoids oxidative damage, phenols involved in defense mechanism, and catechin, also an antioxidant. Given these important products and its relevance in the economic chain due to spirits production, maintaining an elite and conserved quality of this plant is fundamental. This can be reached through micropropagation techniques, which help select one plant with particular desired traits and generate many clones of it in a short period of time in a small space. In this way, we had the main objective of designing and implementing a micropropagation protocol specific to agave tequilana, first by developing a successful cleaning method for ornamental specimens, testing shoot induction in media with different proportions of auxin in combination with cytokinin, and evaluating different light types on shoot development. Also, we pretended to evaluate the cytotoxic effect of an extract from this plant into cancerous cell line HCT116 by analyzing the absorbance morphology and confluence after applying the extract. All this so that we could provide our training partner WeClone with a cleaning protocol that described the steps to be followed to obtain clean explants and a proposal of an enhanced shoot induction media for further testing. We hypothesized that since agaves are plants that harbor a great amount of microorganisms, only an aggressive cleaning protocol with fungicides and antibiotics would work to obtain clean explants. Also, the experimental design was based on the hypothesis that shoot formation is affected by the concentration of 2,4-D and by the type of light to which the explants are subjected. Finally, since the extract from agave tequilana was abundant in sugars, it was expected that it would promote the proliferation of cells. To design the shoot induction protocol, we proposed a series of media supplemented with 4 different auxin concentrations and 1 constant cytokinin concentration. 4 repetitions per combination were going to be grown under white light, while the same amount would be grown under blue-red light. The impact of each factor in each level was going to be evaluated, as well as the impact of the combination of factors to determine which treatment developed the most shoots per explant. As for the constant elements, they included a kinetin concentration of 4.7 micromolar, 25 degrees Celsius temperature, media composition, pH, and explant source. Overall, considering both factors and levels, the appropriate statistical analysis to evaluate the number of shoots per explant was a 2-factor factorial design. Meanwhile, to evaluate the cell proliferation of the HCT116 cell line under different concentrations of the agave tequilana extract for a different amount of time exposure, 30, 50, and 70% concentrations of the extract were prepared and put in contact with the cells for 24 and 48 hours. The normal proliferation effect will be evaluated with the negative control, which contained only cells and media. The first cleaning protocol, which only consisted on ethanol and bleach, had no success at all, since all tubes had fungal and bacterial contamination, as well as the phenolic compound release. Taking these results into account, microdin and fungicide treatments were added, and activated charcoal was added to the media, showing better results since 11 out of 32 tubes showed no visible contamination. However, since fungi continued showing up, another fungicide was added to the protocol and time exposure to the first fungicide treatment was increased. However, this time all tubes were contaminated with fungi and bacteria, but mainly bacteria. The last treatment we tried included the addition of a cleaning stage with copper sulfate, more time exposure to both fungicides, and the addition of ampicillin. With it, we got apparently clean explants. Agave tequilana has many common diseases on the field, but one that we identified grew in tubes was a fungal infection caused by Fusarium oxyporum. Also, from the endogenous symbiotic bacteria, we could detect the tubes that had protobacteria and firmicutes. Meanwhile, uromycetes was the most prevalent fungi. We had multiple findings with each protocol. For the first one, we found that fungi and bacteria were resistant enough to bleach and alcohol, so specific fungi and bacteria killers were added. By using them, we had better results, but still got contamination. Then the following protocol had the complete contamination, which was awarded to possible infection acquired from the laboratory they were stored in, and the fact that plants had been cut for a longer period of time. Finally, by widely increasing the amount of time of the fungicide treatment, we got rid of all fungal and apparently bacterial contamination. However, a white opaque halo was present in all the tubes around the explant. Nonetheless, no compelling results regarding shoot formation could be obtained since the first tested cleaning protocols proved inadequate for achieving a sufficient level of cleanliness in the explants. Although 1 week after inoculating the explants treated with the last cleaning protocol, apparent shoots started to emerge. However, if they are indeed shoots, we must wait longer to observe a more adequate morphology like those reported in the literature, which already have a length of 2 to 6 centimeters and hence properly perform a statistical analysis. Therefore, the effectiveness of the cleaning protocols were tested using a two-proportion statistical test, being those the number of clean explants according to the total repetitions of the two cleaning protocols that were tested, which were number 2 and 4. The most important variables involved were the time exposure to the fungicide and the implementation of the antibiotic. No interaction between them would be evaluated since both are intended to eliminate different components, fungi and bacteria respectively. However, the presence of contamination was evaluated as a wholesome. So, considering that the halo present in the tubes with the last cleaning protocol was due to natural plant expels like meat, we compared cleaning protocols number 2 and 4. The null hypothesis corresponded to that saying that both proportions were the same, while the alternative hypothesis was that saying that both proportions were different. Since the p-value was of 0, less than the 0.05 level of significance, the null hypothesis was rejected at a 95% confidence level, so the alternative hypothesis was true. Then the sample P of the first proportion was of 0.34, meaning that this cleaning protocol had a 34% of success, while the sample P of the fourth cleaning method was of 1, meaning that it had a 100% success rate. On the other side, the absorbance values of HCT 116 cell wells increased accordingly to the extra concentration and time exposure. The half maximal inhibitory concentrations for the 24 and 48 exposure was negative. Although this result can make sense because according to literature, these methanolic extracts contain a high amount of sugars and thus might help cell proliferation cause of the Warburg effect, a negative IC50 result is not reliable, not reported in literature. Hence, a pure bioseparation of the compound of interest must be done and the cells must be subjected to lower concentrations of it to avoid interferences. Overall, throughout these 10 weeks, we found out that agave tequilana possesses high amounts of fungi and bacteria, which need aggressive fungicide and bactericide treatments to obtain clean explants. Then, the final results for the cytotoxic assay were not reliable, but its sugar content might promote proliferation. The cutting-edge biotechnological elements presented in the first experiment proposal were the kinetin and 2,4-D hormone combinations, since they have not been reported to the best of our knowledge in developing shoots for agave tequilana. Also, the use of blue-red lights to agave growth has been minimally studied, but according to Ramirez Mosqueda and team, it is a good alternative that promises to improve the growth rate, so it must be further analyzed in combination with the aforesaid hormones. As for the cleaning protocol, none of the ones used by other researchers up to our knowledge included a simultaneous use of fungal and antibiotic treatment, which promises to be aggressive enough to kill all the pathogens of these plants and create axenic cultures. These proposals considered the market niche in this area. First, the fact that agave tequilana is a highly contaminated plant that is very hard to clean. Also, it is a very demanded plant for spirits production. So, cultivators are in constant need for healthy plants that possess the elite quality for their tequila. In this sense, providing them with the required pathogen-free in vitro plants that have the same superior genotype will allow them to increase and ameliorate their production. The added value of this biotechnological product consists on the provision of axenic and controlled pathogen-free cultures of a single variety of agave tequilana. Some of the advantages rely on the use of resources, since once the disinfection and propagation protocol has been proven efficient, many plants can be produced from one. Also, micropropagation helps in the conservation of endangered species, since several specimens can be produced in vitro and avoid the extraction of wild plants for exploitation. However, disadvantages of this project include the generation of a successful cleaning protocol, which takes up a lot of resources both in terms of media but specifically in plants. Likewise, it promotes the production of monocultures and the use of large spaces of land for the growth of agave, impacting the biodiversity of fauna and flora of the area. Overall, to discard any bacterial contamination in the explants, it is recommended to perform a culture and an antibiogram so that it can be discarded, or in lieu of thereof, find an adequate antibiotic to get rid of these bacteria. Also, the explants that we have up until now have to be incubated for a longer period of time with constant evaluations to confirm that the structures that emerge 8 days after inoculation are in fact shoots. Also, a blue-red light treatment is suggested to be evaluated on the shoot development once the presence of shoots has been confirmed to be possible after the cleaning treatment. Finally, another cytotoxic assay must be done with a pure bioextract at a lower concentrations to obtain an IC50 compelling result.